Hello, everyone. For anyone who is new here, my name is Robert Driver. I'm a postdoc at Duke University, and I want to welcome you to the AOS Student Research Grant Seminar Series here. And just a brief recap, each year the AOS offers funding to students and early career researchers with the AOS Student Research Grant. And the AOS Student Research Grant Seminar Series, which is where we are right now, celebrates the challenging and innovative work of the AOS Student Research Grant recipients by sharing this AOS-funded work with you guys, the AOS members. So each week, we showcase the research and research plans of a 2024, 2023, or 2022 AOS Student Research Grant winner in a seminar-style format. And we have one seminar each week for 15 weeks. We're a little over halfway through now, going until December 15th. And I want to thank everyone for being here. Thank the AOS and AOS Student Affairs Committee, Chris Ann Handel, Ramona Flesher for their support and input, and especially thank Juliana, our speaker today. And before we turn our attention to Juliana's research, I want to mention that if you have questions, there will be time for a Q&A session at the end of Juliana's talk. You can participate by raising your hand, the raising hand icon, and then I can unmute you if you would like to speak. Or you can type your question anytime in the chat, or you can also type your question in the Q&A box. And I will monitor these three ways of asking questions if everybody can remember all those. And I'd like to remind everyone this is an AOS sponsored event and that all participants are required to follow the AOS virtual meeting event code of professional conduct. And I'll post a link to that code of conduct in the chat in a moment. And I will also post a link to a post-seminar evaluation form, which you can fill out to provide us feedback to help us improve the seminar series week to week as we go on here, as well as a contact email for the AOS Student Affairs Committee. And finally, please also be aware that we are recording this talk. Okay, so today, Juliana Villa is currently a PhD student in environmental science and public policy at George Mason University. And Juliana's academic career began with a Bachelor's of Science in Biology from University of Antioquia in Colombia. And following undergraduate, Juliana performed research monitoring the influence of the Ituanga hydroelectric project on bird populations in Colombia, which I thought was very cool. And in 2021, Juliana began her PhD in the lab of Dr. Daniel Hanley, at George Mason University, studying the evolutionary oology, the evolutionary study of eggs of the cowbird brood parasitism system. And Juliana's PhD research on cowbirds is published in PLOS Biology, Biology Letters, Journal of Experimental Biology, and Frontiers in Ecology and Evolution. And so with that, I would like to turn it over for Juliana for her talk entitled Patterns of Collective Host Selection in the Brown-Headed Cowbird System.
All right. Thank you. Thank you for that amazing introduction. As Rob said, yes, I started my academic career at University of Antioquia in Colombia. So, oh, wait.
Looks good.
Okay, there you go.
Yep.
So just to share some pictures of my work there. Yes, I graduated in 2020 in the full peak of the pandemic. And by that time, I started working with animal behavior, specifically using the shiny cowbird. So my work as a behavioral ecologist that studies avian brood parasitism starts over there in Colombia. And then I got the opportunity to meet Dr. Daniel Hanley toward the end of my undergrad studies. And then I came to the US to start doing some research with a different species, the brown-headed cowbird, here in North America. So I've been in George Mason University in Virginia since 2021. Yeah, so really happy to be here, and thank you so much for providing this space. So in terms of what we're going to touch on, I would like to give a general introduction about my research and the topics that are central to it. Then I will switch into two main parts. One is where I'm going to explain about continent-wide patterns of eggshell variation in the brown-headed cowbird, but also I will give an explanation about more like local variation in the brown-headed cowbird. And then I would like to have a wrap up and a takeaway message for the presentation. All right, so let's dig into the topic. So yeah, as I said, I study avian brood parasitism. This is a behavior that has been really exciting for a lot of people. Darwin was one of those, and it's just unique in the sense that in this behavior, there's one bird species. This is, for example, the common cuckoo, the bird that you see here. They don't build a nest. They don't provide any parental care to their offspring. They just go around and find the nests of other bird species. For example, this wagtail. So the cuckoo goes and lays an egg in the nest of that wagtail. And if this bird accepts that a cuckoo egg, they have to raise the cuckoo chick. So it is amazing, but it can also be a costly behavior for the host. So in this image, you can see that sometimes in the case of the cuckoo, the image in the left, these cuckoo chicks come they come programmed to reject the eggs of the host, just completely like remove them, put them out of the nest. So that's very costly for the host. In other cases, as in cowbirds, the parasitic chick can live with the other nestmates, and they share the nest. But this is still very costly because the cowbird chick is going to grow faster and is going to be bigger than the host nestlings in many of the cases. So because this behavior can have implications in the fitness of the host, there are some defenses that have been adapted by the host species. And one of those, one of very crucial defense is egg rejection of the parasitic eggs. So here you can see a host of the cuckoo puncturing one of the cuckoo eggs and just like ejecting it from the nest. And you can see also catbird rejecting that's actually one of my fake eggs that I use in the field, but they grasp and then they remove that egg from their nest. And this behavior is just fascinating because it just starts a whole cycle of evolutionary exchange between the host and the parasite. To be more specific, when the host rejects parasitic eggs that are more similar in respect to their own eggs, they are just eliminating the worst mimicking eggs in the parasite population. That means that they are favoring those eggshells that are more mimetic and that are expressing better eggshell mimicry relative to their own. So egg rejections by the host are the driver for mimicry in the parasite. And in turn, those hosts that are better at rejecting are going to be more successful. So this escalates into a coevolutionary arms races where the host and the parasite are adapting ever improving strategies against each other. So this is a cycle that goes all the way around and back and forth over time. In fact, the cuckoo is this example, is the poster child for coevolutionary arms races and perfect eggshell mimicry. And in this image, you can see the top row is the cuckoo eggs and the bottom row is the host. So you can see that for the most part, for this one single species that parasitizes other host species, it's a pretty good mimicry by the cuckoo. With one exception, you can tell that there is the Danock egg in the middle that is blue. And it's because the Danock doesn't have egg rejection skills. So if there's no egg rejection, there's no mimicry. So this is a good example of how that goes. Now let's switch to cowbird parasitism because the cuckoo is one of the almost 100 avian brood parasites, and there are more things going on in this behavior. So cowbirds, very similar story. We have a female that needs to lay their many eggs. She goes around, find the nest. So for example, a dark-eyed junco parasitizes that nest, and if the junco accepts, so they have to take care of the parasitic chick. What is very interesting about cowbirds in comparison with cuckoos is that cuckoos go and locally parasitize usually one species of host, and the cowbird goes and parasitizes a cardinal within one single community, but they can also parasitize a wood thrush, but they can also parasitize a ton more species in the same locality. So they are targeting all the nests that are around and are possible to parasitize. And that makes things a little more complicated in comparison to cuckoos. And when we see what happens with cowbird eggshells is that you cannot really say that they are mimetic or not. That perfect and imperfect eggshell mimicry is not something you can put in the example of the cowbirds. Sometimes they're going to be a good match for some of their hosts. Sometimes they are not going to be a good match. But cowbirds and cuckoos do have something in common. And this is what I'm trying to explore. We propose that host selection is something that both systems can have. Some cues about this that can indicate that they do have host selection. These are the results for a meta-analysis that look into what are the eggshell traits that are involved in egg rejection responses by hosts. So in the y-axis, you can see different traits, color, maculation, material, etc. And at the top, you can see that eggshell coloration for cuculus, which is the cuckoos, and molothrus, which is the cowbirds, both have a pretty significant and strong effect size, which means that in both systems, eggshell coloration is involved in the decisions of the hosts. But my question is, if this is true for cuckoos, so how is host selection playing a role in cowbirds? And yeah, the task is how cowbirds across the entire distribution in North America evolve their eggshells, how evolution happens in their eggshells when they are coming across all these many species. They parasitize in their entire distribution more than 200 species. So it gets into this mosaic of evolution and interactions that we want to figure out. So that's my question, how coevolution is operating between the cowbird and its many host species. We have a hint into that answer, and we're going to frame it as a different alternative to pairwise coevolution. So pairwise coevolution is the dynamic underlying coevolutionary arms races and these interactions that I showed between a cuckoo and a host species. We're going to talk about diffuse coevolution, where it can be a more appropriate way to describe the interactions between cowbirds and their many hosts. So diffuse coevolution is defined as the coevolutionary exchanges between one species and a host community, or between two communities. So here we're talking about multiple species interacting against the parasite, or in other cases, different to this example, a community co-evolving against another community. So we already had some preliminary studies that suggest that this can be happening for the cowbird, that the selection of cowbird eggshells are driven by the host collectively imposing that selection pressure on the cowbird eggshells, more than just the pairwise interactions between them. So I'm using the framework of diffuse coevolution to explore the interactions between cowbirds and their host, and also to explore how host selection plays a role in the evolution of cowbird eggshells. So as I mentioned before, I will be first digging into a continent-wide project and then a local-wide project. The AOS was involved in both in some way. I got a good match between them. The AOS was intervening more in my local approach that is in the field, but there was also like some funding that went to my continent-wide project. So let's start with the continent-wide project. So as I said, that the hypothesis that we want to test is that host selection in the cowbird eggshell is detected at the host community level. That means that we need to take the complexity of the system as a whole, as each host community is interacting with populations of the cowbird. And this is where I consider that the mosaic gets important because, yeah, the cowbird is widespread in the United States. Here you can see the distribution. So an approach we're taking is dividing the entire distribution of the cowbird into these comparable hexagons. That is something that we need to improve and just have a different framing and try ecoregions. So yeah, just heads up, this is work in progress. So yeah, we want to test how that host selection is more like from the community than from individual host species. And we have 3 different predictions around that, if this host selection is happening by the community. The first one is that cowbird eggshells will differ geographically, which means that those cowbirds that are more apart between each other in geographic distance will be more different between each other. A second prediction is that host communities that are more diverse in eggshell appearances will select for a cowbird eggshell that is also very different, because the composition of the eggshell colors are going to make the comparisons for selecting against or in benefit of specific cowbird eggshells differently. So more diverse and more different host communities, then more different the cowbird eggshells are going to be. And the third prediction is around a specifically diffuse coevolution, where we are going to test, and this is the part that is still in progress. We want to test if actually the different selection pressures by the host community can predict a cowbird eggshell, and if that prediction of the cowbird eggshell is something that we compare and identify as identical to the actual cowbird eggs that we found in museums and in the field. All right, so let's jump into data collection. We have collected a lot of data on eggshell coloration. We go to museums, we grab all the drawers, and we find all the clutches that are parasitized by cowbirds. I went to 3 different museums. This is a picture at the Delaware Museum of Nature and Science. This is we also went to the Western Foundation, one of the most important ones in terms of eggshells here in the US. And we also collected all data from the Smithsonian Eggshell Collection. And then after we got all that eggshell coloration information, so we assign each specimen has a coordinate. So we assign each specimen into one of those hexagons that we have in the grid of the distribution of the brown-haired cowbird. All right. So that data is there, is assigned very nicely. And now I really need to explain how we studied that eggshell coloration that we captured from the field. So bear with me, this is complicated, but not so much, but it's important. So we need to use how birds perceive color, because we perceive color very different to what they do. So for example, we have a clutch of a catbird. This is an egg of a catbird, and the egg of the parasite, the cowbird. We're going to use visual models to calculate how birds are perceiving those 2 colors. We use a package in ARC that is called Pavo. And using Pavo, we're going to include all the photoreceptors that birds have. So we have 3 photoreceptors, birds have 4. So we have the short, medium, and long wavelength sensitive photoreceptor. Birds also have the ultraviolet, and this is why we really need to use this approach. And these 4 photoreceptors can be placed into a color space, and each of the points that form that space is going to be one of the photoreceptors. So that will look like this. So because you have 4, so you can have a tetrahedron, and inside that tetrahedron, you can measure the distances between those 2 eggs, but you can also put inside that space basically anything. And then I'm going to introduce one of the metrics we use the most here, that is called just noticeable differences. So yeah, we have this approach, and to explain what is a just noticeable differences, when we want to compare, for example, those 2 eggs, for our vision, they are different, and for birds, they are probably different too. So we're going to say that those 2 eggs, the white with brown dots and the green one, are noticeably different, then the GNDs are more than 1. And in case that they are unnoticeable, they are just looking identical like each other. So the GNDs are less than 1. And then we can use those differences in color, but also in brightness. So all bird species, like if you take them all and their colorations, you can describe their color to be anything between a blue-green pigmentation and then a brown pigmentation. And then you can have variation of darker and lighter colors. So within this space that we're using of chromatic of color, just noticeable differences and brightness, or achromatic, just noticeable difference, you can basically plot all the eggshells of all birds, including the ones that we're interested in. So we're using this approach. And then I'm just going to jump into our predictions and how we are testing this. So just to remind, prediction number one is that those cowbird eggshells would be different across different geographic distances. So more apart from each other, more different they are. We're using the just noticeable difference between all the cowbird eggs that we collected data for, and we're comparing that with all the distances of the geographical distances between all the cowbird eggs. So we're comparing two matrices, and the way to test if they are significantly correlated or not is using a Mantel's test. Then for prediction number two, we use a very similar approach, but instead of comparing every cowbird against each other, we're comparing every host community against each other. So instead of the cowbirds, we're going to have the hexagons. So we compare per hexagon the just noticeable difference of the cowbird eggshells in both color and brightness. And then the second matrix is the just noticeable difference among the host eggs. So we compare the two just to see if they are correlated or not. And we also perform a Mantel's test. Prediction number two is what I told you about the diffuse coevolution model, where we want to check if the selection pressure can predict cowbird eggshells. And what is selection pressure? Well, it's a number of factors together that influence the success of a cowbird eggshell or of a cowbird individual just that is parasitizing. So the first factor is the host eggshell colors. That is important because cowbirds get compared against the host eggshell. So if they are blue, they're going to look more distinctive compared to a cowbird eggshell than than an egg that is just white or very similar to a cowbird egg. Factor number two is the relative species abundance. How abundant is each species that is getting parasitized, because that influences also the amount of nests of each species in the community. Factor number three is what are the rejection skills, every host. And the fourth factor is how frequently these bird species get parasitized. So our model predicts that within, for example, a host community that has 4 different species, if only one is exerting selection pressure, then that cowbird eggshell can look a lot more like that host species. But then if we have another species that is also having the selection pressure, so that cowbird eggshell is going to move a little in the color space just to be intermediate between those two. And then if you start adding species, so that can get pulled in one way or another, and even just like be the center in the color space we're studying. So just to give an update on that, we are collecting still the data for this. This requires a lot of review of eBird, because we need to build these host communities across the distribution of the cowbird. That's done, but the most challenging part is collecting data on what are the parasitism rates across the all United States or North America for each host species, and we have plenty of them. So we're working on it. So I would like to just present the results we have for the other parts. Just preliminary results in terms of the sampling. Here I'm going to show you only the results for one subspecies of the brownhead cowbird, that is Ater, which is the green area of the map. For that subspecies, we got a little more than 1,300 eggshells measured in the museums. From those eggshells, 479 were cowbird eggshells. This sampling was in 76 host species, and if you summarize it, you have a little more than 300 parasitized clutches just for this subspecies. We do have more data on the others, but let's just start for the one that we have the most data for. And we did excluded some host communities that didn't have enough sampling, and from 30 something, we just selected these 14 that had enough for all of them. All right, so just a quick preview. The this is important because we have thought for the longest time that cowbird eggshells are just not very interesting. They are just very genetic. They don't change that much, and they probably have very little variation. So in this plot, what I'm showing is those 500 cowbird eggshells in that space that I explained of just noticeable difference in color and brightness. And what you're seeing there is black and blue dots. The blue dots are the eggshells that are perceived as identical to each other. The rest of the dots are just completely different compared to the average cowbird eggshell. So here's just to show that they're actually very different when you just consider the whole distribution of the cowbird and the historic records that we have from museums. So we have that, yeah, that 20% are very different in color, and this is an important note. Only color is giving, only 20% is giving differences in color. 82% of those cowbird eggshells are different in luminance. So luminance or the amount of light in a cowbird eggshell has a lot more variation than color. But in the color space, 85% of these cowbird eggshells are just very different to the reference. They are just noticeably different to what I'm comparing to, and those results are significant. I put the test just at the bottom. All right. That just to show, because if you don't have variation in a trade, you cannot proceed with your predictions. But wait, well, I have something to hang on. So yeah, let's dig into prediction number one, where I wanted to see if cowbirds do have variation like in a geographical scale. And I found something very similar to what I was expecting in terms of the variation of the trade, where color is not actually changing a lot for cowbird eggshells. And that makes sense because cowbird eggshells are white. They don't have a lot of pigment. What is very interesting is they do change in their brightness. And they are white eggshells that have brown spots. So that's hint number one. Maybe maculation might be important. And we're seeing differences in brightness. And we're seeing that those differences are significant. And that actually cowbird populations away from each other are more different than the ones that are closer to each other. Prediction number two is tied to the host selection pressure, where we want to see if those host communities are closer to each other, are being parasitized by cowbirds that are more similar between each other. And we found something very, very similar to my previous finding, that is that color in the left is not giving us any signal. But brightness is. So we're seeing some patterns derived from the brightness of the eggshell colorations. And here is very important just to notice that, just to dig a little bit in the discussion, that this luminance can be under that host selection pressure, since we are seeing pretty consistent patterns of brightness being changing over different scales. And the next step linked to that is to check for maculation. We want to know if that light, the brightness and the level of how dark or how light they are in color is related to the maculation patterns, because cowbird eggshells have lots of little spots. So maybe the density of the spots are influencing how bright or how dark they are. So we want to investigate that. And the next step is also just figure out if diffuse coevolution is a model that we can use to understand this mosaic of the cowbird interaction. So yeah, we do need to keep exploring how egg rejection behavior, parasitism rate, and other patterns of host composition and diversity influence the interactions. All right, so let's change gears to more like the local test that we did. This was a local test for diffuse coevolution, where instead of doing the literature review to get egg rejection rates and all of it, we just like got it ourselves in the field. So we're following the same procedure. We're studying diffuse coevolution, but this time in a local community, where we have different host species. We just go and monitor all of them. The hypothesis that we have here is that cowbird eggshell is something that we can predict by the selection pressures of the host community. And those selection pressures are going to be, again, the eggshell coloration of the host, their egg rejection abilities, their abundances, and the rate at which they are parasitized. So for the methods, yeah, we went to the field, we go and monitor nests. We find the nest, and whenever the clutch is completed, so then we can proceed to run something that is called an egg rejection experiment. So I'm going to explain that with more detail. But the field experiments that we conducted were in Boise, Virginia. And the main experiments were in 2022 and 2023. And in 2024, we just like did some additional supplementary experiments to back up our first experiment. So let's check what is an egg rejection experiment. So I was the cowbird. What I did is like I made these cowbird eggshells or cowbird eggs using 3D prints, and I paint them and I made them like all equal. So I went to the field and I say, hey, Robin, you're getting parasitized by me now, and I want to know what you think about this and what is your decision. So I came back 3 days later and determined whether the response to that experiment was rejection or acceptance. Is rejection if that fake egg is gone, or is acceptance if it's still there? All right, so I recorded my response, and after that, I measured the color of those rubbing eggshells. So I use an spectrophotometer. That's the device that just like lights, it has a light that hits a surface. The surface that is the eggshell is going to absorb some of that light, and the other light gets reflected, and that's how you measure reflectance, right? So that's how we measure eggshell coloration. And we did this for the 6 bird species that we were monitoring. That's the great catbird, American robin, wood thrush, the northern mockingbird, brown thrasher, and the northern cardinal. So yeah, all the sampling happened between May and July every day, just going for all the nests and yeah, seeing what was happening all the time. And just to confirm, this is what we did in 2024, where we wanted to know if those 3 days that I allowed were enough. So in 2024, we did the same, just all the monitoring of the bird nests and getting estimates whether 3 days was enough or if birds needed more like 6 days. So we ran experiments. We did like all the monitoring from 1 to 6 days and see if they are responding by day 3 or if they're taking more days. So we did the same. And what we saw is that only 2 birds in the entire sampling we had were like, well, maybe I need 1 or 2 more days. So you can see that one individual, the brown thrasher, and one individual of the northern mockingbird needed more time, but this was not going to influence our procedure. So we were confident that 3 days was enough. All right, so let's switch again to the complicated stuff, but I already explained that. So we already know that that's how we analyze eggshell color. And very similar to what I did before, instead of putting like all the eggshells of all birds here, I'm just using the host community that I'm interested in. And all the comparisons I'm making is within this color space of just noticeable differences. And again, very similar to diffuse coevolution, as I explained before, I'm applying this model to the host community in Virginia here. So I have my 6 species, each of them have their eggshell colorations, each of them are different in their relative species abundances, each of them get parasitized differently. And then I want to apply that model to see if I can actually predict how the cowbird eggshell should look like. Okay, so for the results, this is going to get busy very soon, so bear with me. But so I'm going to show the color space that we have, and these are the eggshell colorations for the 158 egg rejection experiments we run. And then in that plot, you can see a black dot just like right beneath the red dot. The black dot is the actual colors of the cowbirds, and the red dot is our prediction. So I want to zoom in a little bit into that. But we're basically finding that our threshold is like from 1 GND to -1. in both axes. So anything that is like within that -1 to 1 GND is not noticeable as different. They are just identical. So what we found is that we were able to predict using the selection pressure by the host community how the cowbird eggshell would look like. And yeah, we found that was great. It was working. We found a lot of variation in our species. We found that the rejection rates were different. Cowbirds usually reject, as well as American robins and brown thrashers. We found that the northern mockingbird is intermediate, and the wood thrush accepts everything. The northern cardinal was rejecting a little more, but it's still very low. All these rejection rates are non-use. This is what we usually see in like birds all around in North America. What is very interesting is like the stars that I just put on the wood thrush and the northern cardinal. Only those two bird species got parasitized. So here's the main conclusion. Like, even though we were able to use this model of diffuse coevolution for our host community, the only species that is actually exerting a good selection pressure or significant selection pressure on the cowbird eggshell is the northern cardinal, because it's the only one that gets parasitized and is the only one that to some extent rejects cowbird eggshells. The wood thrush does get parasitized, but doesn't reject. So that's not pulling the cowbird actually in any direction. So as part of the discussion for this is that, yes, we were able to predict that cowbird eggshell phenotype using the model of diffuse coevolution, but it requires some caution and just like, wait, we need to analyze and interpret this as it should be, because the cardinal is the only host species that is affecting how the cowbird eggshells are looking like. And we also found that, interestingly, cowbirds and cardinals look a lot like each other in their eggshells. And this can be, we cannot say it's perfect eggshell mimicry between them. We cannot say they are co-evolving with each other. We could say that maybe is a pretty good coincidence, and that the cowbirds are using effectively their eggshell coloration to exploit a bird like the cardinal. But there's a lot more to discuss in this, and well, I will be publishing this paper soon-ish. So yeah, we will see how the discussion goes around with that. But it's just exciting to have a model that can help you put together information from an entire host community and maybe dissect and understand a little more what are the interactions and how they're looking like. Now, so all of this is great. Eggshell coloration, it changes in geography, yeah, there's a lot of variation in communities, but why is identifying host selection in cowbirds important? And this is a great question, because whenever I come across people that talk about cowbirds, I do find this conception that the cowbirds were first distributed in the Great Plains of North America. And after the European settlement, they start just like distributing all across the United States and Canada. And they are just getting to those hosts that are new, naive, and don't know what to do with a cowbird egg. So this creates some perception of the cowbird that makes it complicated, because we do think that egg rejection is not a strategy of many host species, and we tend to believe that they are defendless. I do want to say that host selection in cowbirds can happen, and we are ignoring a very important piece of evidence that has been there, yeah, and is that cowbirds were distributed in North America more widely than what we thought before. So here what I'm showing is just fossils from where? From late Pleistocene. That shows that they were around, and they are not, it's not just that they were around, they were probably also more abundant. And we also know that there were other cowbird species in North America. They went extinct after the megafauna overkill. And this is very important because we already know for other, even for cowbird hosts, but other host species, that once they adapt egg rejection, they can retain egg rejection even if they are not getting parasitized. So my invitation is like, okay, we could describe host selection and try to understand better how this influencing evolution if we just like can be more open to other sources of evidence. And I want to build up on that. And it's like, how is ignoring the different hypotheses about the distribution of the cowbirds important? Like, is that influencing anything at all? Here is a class project that I had for my PhD, where I just like went through all the host species of the cowbird and identified the ones that were affected by cowbird parasitism, and I just picked the ones that have conservation status that can be more interest, or it requires more attention. And this is a trend. We know this really well for the Kirtland warbler, since this is one of the most important conservation priorities in North American ornithology. What we see for all of them is that even though cowbird parasitism is important and meaningful and is probably contributing to the declining of this host species, habitat loss is still the main cause of the decline of those bird species. So for the Kirtland warbler, the population didn't start growing after habitat was restored, even though they kill a lot of the cowbirds. So removing the cowbirds can help just to like don't bring the host species to extinction, but we also need the habitat restoration and just to provide where to nest, and the nesting is actually being successful. So I just wanted to link that, like we still need to do a lot to understand this, and to understand like how host and the integrity of host communities, how that integrity is involved into the behavior. All right, so just to wrap up, I have some main takeaways. With my research, looking to go beyond some classical assumptions. The first one is I want to go beyond coevolutionary arms races and just embrace the complexity of multi-host systems. And I also want to go beyond the expected outcomes. We are all the time expecting to find perfect eggshell mimicry and these nice matches, not just in cowbird parasitism and avian brood parasites, but in any other kind. We want to know the insect that looks like a plant or like a flower, but that is not achievable in every system. And we just not need to like embrace that complexity, and cowbird parasitism is a great example. It does require a lot of probably someone's career. Me, I don't know if I'll achieve that, but maybe. And yeah, with that, I just want to wrap up and say thank you to you for being here, just watching the YouTube video afterwards, and all the people that had contributed to my research in some way, including the US. Okay, thank you.
All right, Juliana, thank you so much for the great talk. I hope people thought more about cowbird eggshell color than they have in the past. I know I sure did. And we will get to some questions. So, Sylvia Halkin, I'm going to allow you to talk. I have allowed you to talk.
Hear me now. Yes.
We can, yes.
Okay. I study cardinals, and cardinals have a fair amount of variation in their eggshells, but they're also probably one of the birds that's least threatened by cowbirds because their young are about the same size. So it's sort of a one-to-one trade-off. And nestling cowbirds look incredibly like nestling cardinals. You have to sort of wait till they're older, and then you can see if they have a bit of a crest, and there's something about the mouth flanges that may be a little different. So I'm just wondering what your thoughts are on that. It might just be that, cowbirds were lucky and initially had eggs that looked like cardinal eggs. Species that they threaten more, of course, the little warblers and such. But yeah, I'm wondering what your thoughts are. And this is fantastic. I'm really glad you're doing this research. It's great.
Thank you, Sylvia. Yes. So cardinals are fascinating. I do have another project on cardinals and just trying to understand like how if they reject, how what are the cues they're using for doing that. We also have Dr. Virginia Renati and Brian Peer, who tried to understand if ultraviolet light was related to the ability of cowbirds of cardinals to reject cowbird
Mhm.
eggs. But yeah, we do know that cardinals are just a great host. They are bigger. They have a bigger nest where they can have both. And there is a study, a study that I really like. Eckerly, I think, is the author, where he has a pretty good sampling, and he has nests of cardinals parasitized and non-parasitized, and he finds that that it seems like cardinals do just well when they get parasitized by cowbirds, which is interesting. I'm like, okay, but still, like, you are accepting and sometimes rejecting, and I see behaviors that are like, okay, you're burying, like, you're pushing down the cowbird eggs into the
Mhm.
lining of your nest. So that means that you have adapted some rejection skills. So
Mhm.
I'm between, like, okay, there is maybe some cardinals can deal well with cowbird parasitism, but there's still some adaptation against cowbirds,
Oh,
because they
yeah.
don't just push down their own eggs. It's just like you go and find the cowbird eggs just like hiding in the lining of the nest. So I am curious, like, a lot, like how can you design an experiment just to go and check what's going on, and if it's actually cowbirds exploiting the coincidence of looking a lot like cardinals in many ways,
Mhm.
because it happens with the chicks, it happens with the eggs. It's just like you are just like using the coincidence, and maybe cardinals and cowbirds were under similar selection pressures, like their ancestral state of the eggshell was like under predation before than under the interaction, and then it happens just to be really effective for cardinals. It can also happen that they just don't care, which I think is what happens also with the wood thrush. Wood thrushes are also like um, big host, and they are nesting in the forest. Maybe they have enough resources. Maybe they can just take care of the cowbirds with no problem. But yeah, I'm very happy to talk with you. Since you study cardinals, I do have many questions.
Sure. Let's rate each other. You can find me on the internet.
Okay,
Thanks.
cool,
And
cool.
I'm one of the authors of the Birds of North America
I
species.
think I met you in Canada.
Yeah, but I am looking forward to your research and to talking more. Thanks. Yeah,
Nice, thank
cardinals
you.
probably started out in the American Southwest, and it probably didn't have as wide a distribution as cowbirds for, the last 150 years, but they're kind of catching up. So that's another factor. That's all.
Yeah,
Thanks.
okay. Thank you.
Great.
All right, great. Thanks so much, Sylvia. And now I will allow Renee Corrado to talk. I've given you permission. But yep,
Okay.
yep,
Oh,
great.
you hear me? Okay.
Mhm.
Hi, Juliana, and that was an awesome presentation. It's nice seeing you again. I have, well, two questions. First, does a host reject eggs by breaking them? If if they do, how do you measure that with your eggs that you put in those nests, because they cannot break those eggs? How do you know that they will reject or not?
Yeah, that's a good question, because some birds, especially the smaller ones, can use the piercing to reject because their beaks are not big enough to grasp. But yeah, my eggshells are not breakable. Like the 3D prints is not something that they can break. So in that case, they will have to remove it through grasping, just like grabbing the egg and removing it, or doing something else. But in my case, like all the bird species that I had were big enough for just performing grasping of the experimental eggs.
Okay. Thank you.
Yeah. Yeah, thank you. Nice to say hi.
And good question. Um, okay, Robert Mulvihill, sorry, asks, what about egg size and shape variability and its possible significance with respect to host acceptance or rejection? Right. So, size and shape in addition to your color analysis.
Yeah. So we've seen a lot of size variation, for example, in the cuckoo. And what is being shown is that eggshell coloration is more of a signal than size is. Now, in terms of shape, there are also experiments. I think Mark Haver has multiple papers, or at least a couple of papers on shape, where certain shapes are just not egg-like. So that does contributes to rejection. Is like if something is not like looking like rounded, it's going to get rejected. But in terms of like how much variation can a cowbird have, I don't think they can be very flexible about it. It's gotta be whatever fits an oviduct of a bird, and that's going to be just pretty much like an eggshell. Now, there are other examples where egg size, it is important, especially for cavity nesters. And here I'm talking about the shiny cowbird, where they do reject whenever that shiny cowbird is just smaller. It gets rejected, and it's just like this tactile cue because vision is not possible within a cavity. I hope that answers your question.
Yes, he just clarifies with the comment here. I mean that cowbird eggs are distinctly less tapered at the small end compared to their host species. So, like, are they more pointed? I believe, Robert, I'm trying to make sure I get your question right.
Yeah, I think I don't have a recollection on like a evidence that shows that shape is relevant in comparisons between host and cowbird eggshells. Yeah, I haven't seen anything that shows that, like how that can influence. But yeah, I will be looking for that type of research.
Right. So I think the background or the motivation for this question from Robert is that he says he studied dark-eyed juncos for more than a
Oh yeah.
decade. Population experienced a significant cowbird parasitism pressure, and the eggs were distinguishable by the distribution of the spotting, which maybe your talk emphasized a good deal. But then he says also by their shape. Sometimes
Okay.
juncos, getting comments. Sometimes juncos buried the cowbird eggs, but more often they accepted them. So it seemed to his eye, the shape was something that jumped out.
Yeah, that's interesting. I think it's interesting that Rob mentioned the distribution of the spotting. So yeah, I don't know if the shape can be a factor, but yeah, spotting, I think it could be that the trait that they're using. But yeah, really curious about that. And it seems that Rob has an excellent data set too on dark-eyed juncos. 10 years sounds pretty good. So yeah, good.
A lot of variables to measure potentially
Yeah.
between the different eggs. So I'm just making sure because we're getting a lot of comments. Rosa Jimenez asks, says, thank you for the amazing talk. And would the parents abandon the nest after being parasitized? Maybe in your local experiments, that is.
Yeah, we did have and actually I didn't include this species just because it felt immoral to do it, but I didn't run experiments in the chipping sparrow. They can just abandon the nest when they get parasitized. That's like another way to respond to that. In my case, I do have supplementary analysis where I'm controlling for abandonment too. That's like my control experiment is checking if I'm the one who is causing abandonment, or I'm the one as a human that intervenes in the nest. I'm causing any rejection that is not linked to just experiment. And I found that no, that I was not causing abandonment, but yeah, chipping sparrows, they do abandon when they get parasitized. And the difference is like this is a smaller bird. So I do wonder about like their ability to actually graft and remove the cowbird egg.
And I think there's some comments in the chat as well as the Q&A. So I just want to figure out which to read here. So Mary Whitfield says, I think egg burial is often a result of mistiming of parasitism. In my experience, if the cowbirds lay their eggs in the nest too soon, for example, before the host lays its first egg, the host will often bury the egg. So
Okay.
did you time your, I guess a question off that, did you time your egg placement for a
Yeah.
particular moment in the clutch size?
Yeah, thank you, Mary. That's a great comment. That makes a lot of sense. Like if you get eggs, like if you're a bird and you get some eggs that are not yours because you know you haven't started laying. So yeah, burying them is the way to do it. What I've seen in shiny cowbirds with a sparrow they parasitize is like whenever they get parasitized before they just start a new nest. It's like, I'm not doing this, I'm going somewhere else. This is not good. And then about the timing of my experiment, yes, I did waited for the clutch to be completed before starting the experiment. Like I know more or less, no, yeah, I know what is the number of eggs to expect from all the species. So I was just like, go for it. And they lay eggs every day. So you just kind of like go, you have 2 eggs that you know that the experiment can be started the next day.
And Sylvia has one more comment, I think, relating to the cardinal discussion before. If the rejection of a cowbird egg allows a cardinal to raise one or more cardinal offspring, natural selection would favor cardinals that reject cowbird eggs, since insect food for nestling cardinals is
Yeah.
labor intensive. Um, and I think a related comment following that up from Sylvia, have you seen any evidence of revenge host nest destruction by cowbirds whose eggs have been rejected? That has been reported, but I don't know how much data there are. So,
Yeah, thank you for the comment on like how the direction of natural selection will play in that case. And yes, yeah, it's still like providing food for a chick that is unrelated offspring. And the revenge, yes, we were running an experiment with fake cowbirds and speakers, and we did see that they can just compete for a cardinal nest, and they I don't know if nest destruction like the nest architecture per se, but they can actually go for and destroy the eggs of the cardinals.
all
All
right.
right.
So we have like a minute or two left. I'll just ask a real quick question. When you go through the eggs in the museum, and some of them are coming from similar locations, are there ever some from the same year? Is it possible that a single female cowbird laid multiple eggs in your data set?
I think it's possible, yeah. Sometimes there's collectors where it's just like a specific field site and they just go and do this. And I seen yeah, I seen that date. I actually haven't thought about it, that pseudo replication for
Hmm.
sure.
Yeah, but there's no way to know, right? No one would ever be even be possible to record that. I would.
No, and yeah, that would be hard. And you also just want to have as much sampling as possible from one
Right,
specific place. So if you just
right.
like start removing, you can know though, you can identify where when the phenotype is just too off from each other. Like you have 2 eggshells and one of them just look very different to the others. Like
Hmm.
this probably coming from 2 different females.
Hmm.
But it can get finicky for sure.
Right.
Yeah.
Yep, gotcha. All right, and
Yeah.
sorry.
Yeah, and
So
then Sylvia does say that they do
yeah,
remove a cardinal before laying their own. Yeah, which
yeah.
is also interesting because it's like do parasites recognize other parasites and the host's eggs? So yeah, cool.
Right. So conversely, the other way
Yeah,
around.
the other way around. Okay.
All right. Well, so many questions. Thank you very much. Great audience participation. And so everybody really enjoyed and responded to Juliana's talk. Great. And so really appreciate Juliana speaking today. Lots of people with the emojis and with comments and everything. But we're at 1 o'clock. Chris Ann has put the seminar feedback form in the chat if you would like to check that out again. We have more questions coming in, but we are up against it.
Okay.
So please Google Juliana or look
Yeah,
up.
I also have my website. Just Google my name and it should show up. It's a Weebly website. But yeah, really happy to get any emails with additional questions and to talk for sure.
Please reach out to Juliana for her research. So thank you so
Okay.
much. All
Thank you
right,
for
we'll
the
see you all.
space. Bye bye,
Bye,
everybody.
guys.